established over that period of allopatry, and also how viable the hybrids are. So in some cases, you have these reproductive boundaries being reinforced, and there's no hybridization between lineages. They continue to evolve, speciate. In other cases, these reproductive boundaries are completely eroded, and you can have lineage fusion. There can also be genetic swamping by the more fit lineage. In some cases where the hybrids are more fit than the parents, there can be selective mating that forms a hybrid taxon. Or you can have a stable hybrid zone. So hybrid zones can take many forms. You can have tension zones where you have primarily hybrids that don't exceed the F1 generation, and they have very small zones of very steep genetic lines. But you can also have hybrid swarms. So in hybrid swarms, there's an erosion of reproductive barriers that allows for extensive introgression. Hybrids are fertile, and there's lots of backcrossing. One of the most recognizable hybrid swarms is in sticklebacks, where the hybrid swarm formed due to an ecological disturbance. But you can also have a hybrid swarm form when two lineages naturally come into contact. So my study organism are the blacktail and the mule deer. And these subspecies diverged during the last glaciation. Blacktails went to the Pacific Northwest, while mule deer shifted their range southward. And about 8,000 years ago, these two lineages came back into contact along the Cascade Ranges. So that's the mountain range that starts in southern Canada, goes to northern California. And a little bit about these animals. Black-tailed deers have black tails. Mule deer do not. Blacktails are also about half to 2/3 the size of their mule deer counterparts. And both of these subspecies have great dispersal abilities, and they can move up to 25 kilometers. So we had two main aims for this project. The first was to compare population structure using mitochondrial DNA, microsatellites, and SNPs in order to get a better grasp on the past and present dynamics of this swarm. And we also wanted to further use the SNPs to explore gene flow across the genome. And specifically, we wanted to quantify the strength of selection on protein code and divergence. To do all of this, we got hunter-harvested samples from across the state of Oregon. We sequenced all of them for the D-loop region of the mitochondrial control region. We also genotyped them at 10 microsatellites. And for the SNPs, we actually did an exon capture project using 3 blacktails and 4 mule deer. And after our filtering process, we identified SNPs that we thought were in genes that may be involved with mate choice. So genes involved with olfaction, those involved with immune function, and we also handpicked SNPs that were really good at splitting into lineages, so ones that were fixed in either one lineage, the other, or both. So we ended up with 95 SNPs that we later screened across the remaining 165 individuals. So here we have our sampling efforts. A lot of these samples were actually taken really close to Portland, which is indicated by that star. And as you can kind of see, we did our sampling in 3 main transects because we thought that there might be some longitudinal differences, but we did not see any significant differences in our data set, so we just pulled all the samples. And we found that indeed, there are 2 main genetic clusters, blacktails and mule deer. And on each of these plots, I've ordered all the individuals from west to east based on their distance to that squiggly line, which is the Cascade Mountains. And that's also indicated by the dashed line on each plot. So for the mitochondrial results, we found that there was 6.4% pairwise sequence divergence between these we found that we end up getting a lot of hybrids, mainly centered around that cascade ridge line. So we considered a hybrid to be anything that had less than 90% probability of being assigned to either of the putative parental lineages. So when we ran some Klein analyses, we found very similar Klein shapes across all markers, with the Klein center occurring just the east of the cascade ridgeline. We also wanted to look at the direction of introgression in this group. So in a lot of mammals, especially deer, we find that females tend to prefer bigger males. So we thought that we'd see this pairing of hybrids: small black-tailed deer female, large mule deer buck. And in reality, we didn't. We actually found that these huge mule deer females were mating with these tiny black-tailed deer just as much as the opposite cross. But we're looking at the hybrids that actually survived. So it could be that we get this pairing a lot more in nature, but the offspring just don't survive because perhaps the doe is just not big enough to carry the hybrid offspring, or there may be some genetic incompatibilities. So what's really perplexing for me is if we had both of these groups of hybrid crosses happening, is there any reproductive isolation? Maybe there just isn't. And that would kind of make sense because deer are mobile. So why would a buck mate with a doe of a different subspecies when he could just travel and find one of his own lineage? But then, if there is no reproductive isolation and they are just interbreeding, why do we still have this hybrid swarm? Why don't we have complete lineage fusion? So, to get to maybe what is preventing lineage fusion, we had another look at our SNPs to see if any particular SNPs and genes were under greater selective pressure. So after we ran BaseScan and PC-Adapt for outlier detection, we found higher selection pressure only on a single SNP, and that was in a gene, the eukaryotic translation initiation factor, gamma-3. Its function is a bit unclear. It seems to be involved with mRNA being directed to the ribosome, and in one study in mice, it was shown to be related to male sterility. So that's kind of cool. But what about all these other SNPs? Maybe, possibly, stronger selection? There was no real rhyme or reason. They were scattered across the genome. They had very different functions. And we were kind of confused because we picked SNPs in genes that we thought would be under higher selection. So the fact that we're not seeing that tells us we're really off the mark. We found, in general, weak selection across the SNPs that we looked at. In some cases, when we ran a Klein analysis on particular SNPs, there was no difference in allele frequency across the hybrid zone. So it's probably a case of a bunch of SNPs having a small effect in maintaining the dynamics of this swarm. So instead of just sitting in the lab, why don't we go back to the ecology? These deer live in very different habitats. We have blacktail so I got these messed up, in Western Oregon, where we have these lush, dense forests, and across the Cascades, it's this very dry scrubland. So one would assume that these populations in very different habitats would be under very different selection pressures. So a next step would be to look at genes associated with different ecological adaptations to see if there's stronger selection pressure on them. So in two lineages, blacktail and mule deer. And despite the fact that they have different habitats, they're very different sizes, and they have high mitochondrial divergence, they can still interbreed. And so we're still trying to get a grasp on what is allowing them to interbreed, but what is also preventing lineage fusion. And that's likely being driven by the ecology. So, as with any project, ours could benefit from more SNPs. Due to our limited genomic resources, we only have the capacity to really look at 95. I would love to look at thousands and do a nice RADSEED project, and also look at SNPs that have varying mutation rates so that we can look at possible changes in the hybrid swarm over time. And it would also be great to extend our sampling range across the entire point of contact to see what those boundaries of the swarm are. And with that, I just want to acknowledge some of our collaborators, particularly Jim Heffelfinger, John Powell, and Don Whitaker, as well as our colleagues at Montana and at University of Wisconsin-Milwaukee and UWM for funding this project. Thank you. Any questions? Yes.
So because these deer are so mobile, do you think that there could just be some sort of, like the hybrids are just moving around, and so it's disrupting your climb analyses? So you're finding them over